Thank you very much. Our next speaker, which will be watching a video from Dr. Bob MacLaren. Dr. MacLaren is a professor of ophthalmology at the University of Oxford. He is a consultant ophthalmologist at the Oxford Eye Hospital and an honorary professor of ophthalmology at the UCL Institute of Ophthalmology. Honorary consultant vitreoretinal surgeon at the Moorfields Eye Hospital and an NIHR senior investigator. Together with the Oxford University in 2014, Dr. MacLaren co-founded Nightstar Therapeutics to expand the retinal gene therapy toward worldwide approval for patients with retinal diseases. After listing on the NASDAQ in 2017, Nightstar Therapeutics was acquired by Biogen in 2019, making it one of the most successful British biotechnology company buyouts of all time. In 2023, he co-founded Beacon Therapeutics together with Syncona Partners and achieving the largest equity at launch for any Oxford spin-off company to date. Over the last decade, he has also raised over $1 billion for funding for retinal genetic therapies.
Thank you very much indeed for inviting me to speak at the CRF meeting. I apologize I'm not there in person, but I'm currently traveling and doing a lot of work in Europe on our gene therapy programs. I had a chance to meet many of you in Montpellier in France last year, and I just wanted to give you an update on where we are with choroideremia and what our current areas of interest are that will enable us hopefully to develop a treatment using gene therapy in future. Now, in 2003, we reported the results of the phase 3 trial using retinal gene therapy to treat choroideremia. This was a multi-center study involving sites in the US, the UK, and Germany. We were able to show in this study in total, we recruited 160 patients into this trial. And within this, patients were allocated to a high dose, low dose, or control group and followed up for 1 year to see the endpoint. The goal had been to improve vision in these patients by at least 3 lines of vision, which was mandated by the FDA. The results showed improvements in vision in patients treated late in the disease, but there were not enough patients achieving 3 lines of vision gain to meet statistical significance for the FDA. In contrast, statistical significance was met for patients having a 2-line gain, and all the other parameters, including preservation of vision within 1 line and the mean change in vision, all showed a greater trend towards the high dose in these patients having gene therapy. So the problem we have is that the treatment effect we've observed is not of great enough magnitude to justify the intervention, according to the trial regulations and the agreement we had for the phase 3 with the FDA. But the data, of course, are very good, and good enough at least not to stop the program completely, but to look at other options of what we can also look at, potentially to show a treatment effect with gene therapy. Now, I've been working in the field of retinal gene therapy for nearly 20 years. Clearly, I'm very positive about it, and I've seen the effects in treating retinal diseases. But running clinical trials is more complicated. Because patients have complicated diseases, the endpoints are difficult, and ultimately, we need to show slowing of degeneration in a disease that's probably the slowest of all retinal degenerations. That, of course, is of great news to anyone with choroideremia, because most will preserve vision for many, many years. But the slow rate of degeneration makes it very difficult for us as clinicians in designing clinical trials. This is an example of surgery in one of our patients in the phase 3 study. And you can see here the virus being delivered into the subretinal space by an injection. But there's really only a very small central area of residual retina from which we're expecting to get a small improvement in visual acuity. It's very difficult, the surgery in these patients, because obviously operating in such a small area risks damaging the retina, which is already very fragile. And also, it should be obvious to you to see that most of the retina has already degenerated, and the surviving retinal pigment epithelium here, where the injection is taking place, is likely to be very diseased, and very diseased by the deficiency of the REP1 protein. And despite some improvement in function following the vector, it's likely that the RPE cells are themselves very diseased and may indeed be past the point of no return in terms of being able to rescue them from further degeneration. And indeed, there is nothing so far that we've seen to indicate that the retinal degeneration will be slowed significantly following gene therapy. So just to summarize, the phase 3 trial was predominantly in very late stage patients in whom the visual acuity had already begun to decline, because we need to have patients with a reduction in the visual acuity to be able to measure any improvement. We have also been looking, however, at patients with very early choroideremia, and we've identified a new feature on the imaging known as the smooth zone. And those of you who follow the literature will see I have several publications on this. Effectively, we have an area in the very center where the autofluorescent signal, rather than being mottled, as it is in most cases, is very, very smooth. We call this a smooth zone. When we switch from autofluorescence to infrared from on the Heidelberg spectralis system, we can see on the infrared image there's preservation of this signal, which represents the melanin granules in healthy retinal pigment epithelial cells. We've therefore classified this into 3 groups. We've got patients with the early stage who have the smooth zone, a preservation of the infrared imaging. We call this group 1. We've also got patients in whom there is some infrared visible signal, but without the smooth zone. And the final stage patients where we may have just a mottled zone, but no infrared imaging showing any signal at all. Groups 1, 2, and 3. And when we look in the microperimetry in these patients, what we notice is that patients who have the microperimetry on the center of the smooth zone, in this area, the retinal function is relatively normal. And if you want to see this in more detail, please look at the paper by Federica Polli et al. With myself as the senior author, looking at the autofluorescence relating to the microperimetry. And we know the function of the cells over the smooth zone is relatively normal, and anatomically they appear to be intact because the OCT scan shows nice preservation of the photoreceptors here, but disruption as we move out here into the mottled zone. And if you want to have more detail of that, please have a look at the paper by Marta Stevanovic, who was working with my team at the time in Oxford, correlating the OCT scans with the smooth zone. So the bottom line is, we have identified in early stages of choroideremia a central area in which the retinal function and the anatomy appears to be normal before the onset of degeneration. So the real question, of course, is what would happen if we were to apply gene therapy in early stages. Clearly, we're not going to get an improvement in visual acuity, because in these patients, the visual acuity was already near maximal at greater than 20/20. And we'd have to wait a long time to see any slowing of the degeneration. Fortunately, in parallel with the Nightstar Biogen trial, we also ran our own investigator-led study in Oxford to assess the effects of gene therapy in choroideremia in very early stage patients to ask this very specific question about slowing of the degeneration. Here is some preliminary data we have after several years of a patient in this trial who happened to have therapy in the treated eye affecting the smooth zone which was present at the time of the intervention. Here you can see above the control eye and below the treated eye. Now, what's interesting is if you map an edge around the edge of the autofluorescence area, you can see a relatively well-preserved area at the beginning and then a gradual decline in both the control eye and the treated eye, showing very, very similar rates. We're looking at shrinkage of the very outer edge of the autofluorescence area. But if you then focus more on the smooth zone, which is just this central part outlined by the yellow dotted line, there does appear to be preservation of the smooth zone in the treated eye compared to the control eye, supporting the hypothesis that we may be able to slow the degeneration by early intervention in choroideremia. Now, as I mentioned at the beginning of this talk, the problem we've got is that this is a very, very slow degeneration, and we have very few patients with the smooth zone in whom we've assessed the gene therapy treatment. So we will need to continue waiting a bit longer in order to get a bigger picture of exactly what's happening. In parallel with this, I'm extremely grateful to the Choroideremia Research Foundation and also Foundation Fighting Blindness and Fight for Sight in the UK for supporting us to conduct some further clinical studies in choroideremia patients to look at the smooth zone in more detail. If we can show a slowing of the degeneration in the smooth zone, we then need to think about ways in which we can present the data to the FDA and argue the case that this is an important clinical endpoint that would be a valuable thing to preserve and might therefore justify approval of the gene therapy treatment. This treatment, however, would be restricted to early stage patients in whom we think the greatest treatment effect would be seen. So my colleagues, other investigators looking at choroideremia in the US and in Europe may well be in contact with patients with choroideremia over the next 6 months to encourage them to come in and have some assessments to see if we can document the smooth zone in more detail in order to find out a little bit more about it and how it may provide us with a useful biomarker to complement the phase 3 trial, which showed improvements in visual acuity, but no obvious slowing of degeneration, with an early stage intervention that clearly cannot show improvement in visual acuity because the visual acuity is already at maximal, but may show slowing of degeneration in an important anatomical landmark that we can detect on the imaging. So finally, I would just like to thank everyone again for your continued support for our research program. And my apologies again, I can't be with you in person this year, but I look forward to catching up with everyone again at some point in the near future. Thank you very much indeed for listening.